Okay, so we're going to see how fast I can teach this because I have about 5 minutes, but I thought it'd be cool to learn about prokaryotic cells. So prokaryotic cells are basically anything that does not have a nucleus or membrane-bound organelles. So we're going to have one compartment and everything gets stored in that one compartment. These guys were the first around, the simplest around. They're the original motto of biology. So we've got some interesting evolution behind prokaryotes. They're going to end up falling in two groups called bacteria and archaea. Now, in biology, whenever we do phylogeny, which is like when we look at how different groups are related to each other, we are going to say that they had a common ancestor that is not around anymore because that common ancestor evolved and split into these two things. So when we're considering the three domains, which are the biggest classification that we have for organisms here, they all have a common ancestor that we call LUCA, last universal common ancestor. And eventually, once upon a time, this split into bacteria and another common ancestor, which is going to be here. And this common ancestor split into archaea and eukaryotes, which are the ones that we are and animals and plants are that have nuclei. So it kind of seems weird that we're more related to archaea than archaea and bacteria are to each other, but there's some cool little things. So like archaea have a cytoskeleton, they have our kind of ribosomes. There's some other things that I can't think of off the top of my head, but they've got some interesting similarities. So if anybody ever asks, bacteria and archaea are not as related to each other as archaea and eukarya are to each other. So if we are getting into the anatomy of a prokaryotic cell, we do not have a nucleus, like I said. We have something else. It is called a nucleoid. It's just a concentrated area where our chromosome lives. And this chromosome is going to be one single chromosome, and it's going to be circular. So they've got a pretty nice, efficient, replicative machinery because they do not have to worry about telomeres and ending the strand and other things that we have to worry about. There are no organelles. The only thing that's kind of close to an organelle is a ribosome, which is the functional unit of translating RNA into protein. They're going to have membranes like we have, proteins like we have, and they're going to have some extra layers on the outside. So you might have a cell wall. Most prokaryotes are going to have a cell wall. And then we've got some appendages that help us move around, which are going to be flagella. That's these long ones. Flagella is Latin for whip, so they're kind of going to go in a whip-like motion and make the bacteria swim. And then these little guys are called fimbriae. They help the bacteria sense things around it and adhere to surfaces. So this is a bacterial cell. We're going to largely ignore archaea, although archaea are really cool. Generally, if you're in a biology class or if you're taking a standardized test like the AP Biology exam or the MCAT, they're going to focus on bacteria. And there's two main kinds. I said times. I don't know what I was trying to say, but there are two main kinds of bacteria, and they go by this stain called the Gram stain. So in the Gram stain, what we do is we have a bunch of bacteria, and we will flood them with a dye called crystal violet. And crystal violet attaches to anybody and everybody in the bacterial world, right? But then we are going to add something called Gram's iodine, and Gram's iodine makes complexes with crystal violet only on this substance called peptidoglycan. And peptidoglycan is on the outside of the cells, the bacterial cells that we call gram-positive. Meanwhile, the gram-negatives, they've got this extra membrane on the outside that's going to protect them from having this complex adhere to them. Once we kind of wash off all the extra crystal violet and all the extra iodine that didn't complex onto peptidoglycan, we are going to counterstain with something called safranin, which is going to turn up pink, and that's going to attach non-differentially to everything that has not already been stained, aka the gram-negatives that have not already been stained with crystal violet and the iodine complex. So this is what gram stain looks like. These are my gram stains that I did with a really cool microscope apparatus that attaches your phone to the lens. I love that. And all the gram-positives are going to end up looking purple, all the gram-negatives are going to end up looking pink, and that is because these gram-positives have this peptidoglycan cell wall layer on the outside of their membrane. Meanwhile, the gram-negatives, like I said, they're going to have an outer membrane, they're also going to have an inner membrane, and then this in here, we call this the periplasmic space. So it's peri means like in between or during, So it is in between the two plasma membranes. It's periplasmic, and in there they're going to have a couple sheets of peptidoglycan, but not as many. So that is basically all that you have to know about prokaryotic life for a basic knowledge for a biology course. So I hope this helped, and I hope you think these gram stains are as pretty as I think they are.